This is the Boring Climate Podcast, a platform where we encourage serious climate conversations. I'm your host, Vishwada. Today, we're trying to unveil the hidden life of one of India's most elusive but favorite wild mammals, the tiger. And to do that, we're speaking to a scientist who uses the tiger's poop to study its past, present, and future and answer the tough question: how does ecological and climate change affect this endangered big cat and other animals around us? Dr. Uma Ramakrishnan leads the molecular ecology lab here at the National Center for Biological Sciences in Bangalore, and her team uses genetic and molecular tools to extract valuable information from poor DNA sources like fecal matter or loose fur and glean insights into some of these tough questions. Her work has wide-ranging application for conservation action and even policy design and planning. Welcome to the show, Ma. Thank you for taking time.
Thanks for having me.
So to start off with, you are a molecular ecologist, and your work here tries to answer a wide spectrum of questions. What are these questions?
So basically, I'm really interested in understanding nature, and from a very young age, I loved animals, and I wanted to understand more about their lives. And sometimes you can watch them, but they still don't reveal their secrets.
Hmm.
And some of these secrets are really critical to conserving their future. So early on, when I was probably in high school, I learned that you can use things like DNA as a source to understand more about animals. The story of our past is written in our DNA, and things that are going on today, like whom we're mating with, and so on. And so DNA is like a cheat.
Hmm.
It allows us to understand a lot about animals, and so I basically took that route of using the lens of genetics and molecules, DNA, some work with chemicals as well, to understand more about animals.
What are some important questions that NCBS is working on in your field as well as others?
So, the NCBS, or the National Center for Biological Sciences, is a center under the Tata Institute of Fundamental Research. And basically, our goal is to understand life,
Mm.
right? Biology, basically. And we have people who work across scale in biology, from people who work at the level of molecules to ecosystems, and theory people who try and make theory for understanding biology. I'm part of the Ecology and Evolution group. We also have an amazing master's program in
Mm.
wildlife biology and conservation. And in the Ecology and Evolution group, there are people who work on understanding the genetics of phenotypes and butterflies, how adaptation happens,
Mm.
um, and also a couple of colleagues who work on vegetation and ecosystem dynamics. So,
Mm.
understanding how, with changes in climate, for example, with drought, how is our vegetation going to respond? And this is, I think, really important in the
Mm.
context of the climate space.
Right. Before we zoom into your actual research, to set context for our listeners, three basic questions. What is molecular ecology? What is DNA? What is a genome and what is a gene?
Great. So, molecular ecology is, like I said, using molecular tools to understand more about ecology, which is basically interactions between animals and themselves, or animals and the environment. Um, what was the second question? What is a
What
genome?
is DNA?
What is DNA?
What is DNA?
DNA is deoxyribose nucleic acid. It is the hereditary material in many species, including from viruses all the way to birds and mammals. So it's basically what traces ancestry. Um, a genome is basically your an instruction manual for how you set up your whole body and everything, and every cell has a nucleus which holds a copy of our DNA or the DNA of any animal. And genes are basically parts of the genome. So, not all of the genome uh, codes for protein, makes something else. Parts of it controls the other parts of it, or we don't really know why it's there.
Mm.
And so genes are that part of the genome which codes for or makes protein stuff in a way.
Mm. Right. To get into your research, what led you to tigers and specifically to trying to understand these elusive creatures just using their genetic identities?
Yeah, so basically, I came back to India. I did my PhD and postdoc in the US, and I was itching to come back and work on Indian wildlife. And when I came back in 2005, I happened to, just before that, I happened to read a paper on tigers, which suggested not very high genetic variation in India. And that made me a little curious about those results. And I happened to interact with and talk to Professor Ullas Karant, who was a famous tiger conservationist, and he was very interested in trying to start work on genetics in
Mm.
tigers. So these were just primers which pushed me in that direction. But also, tigers are a good system because I'm interested in understanding processes that push species towards
Mm.
extinction. Tigers have faced dramatic decline in numbers and habitat fragmentation, and yet unlike, say, maybe a mouse that has faced this, tigers are pretty well studied ecologically. We
Mm.
know quite a lot about them, their breeding, when they breed, how long they live, how far they move, stuff like that,
Mm.
basic stuff, which is important when thinking about what the genetics mean.
Mm.
So they were really an ideal system, and I
Right.
was really lucky to
Mm.
be able to pursue this work.
Right. And how do these genetic tools kind of compare with traditional approaches like camera traps?
Yeah, so camera trapping is, I mean, for tigers, it works really well because they have unique patterns. And what the genetic, the DNA from, say, tiger poop, or as you said, fur, bits of fur, will also give us kind of unique identities of individuals. So they compare well, but there may be some habitats where camera trapping is harder,
Mm.
and genetics might be an only source that is for identifying individuals. Where genetics really kind of surpasses or adds much different information is in looking at things like inbreeding
Mm.
or potential effects, physical defects,
Mm.
which we can track back to specific genes or
Right.
genetic variants. And also in trying to understand connectivity.
Mm.
So while we can use, radio collars and follow animals and get some idea of how far animals are moving, movement tends to be relatively rarer.
Mm.
And so, if we actually genetically sample to regions, depending on whether the variation appears mixed or separate, we can better understand connectivity.
Right. Which brings me to the next question is what do you understand from poop? And would it also not give information about, apart from the tiger's own genes, what it's eaten and how do you
Yeah,
differentiate?
yeah, yeah.
Yeah.
So that, of course, the
Yeah.
poop has lots of kinds of DNA.
Mm.
It has tiger DNA, it has prey DNA, it has bacterial DNA,
Mm.
fungal DNA. So
Mm.
like it's just got tons of DNA,
Right.
right? And in fact, when you just take a bit of poop and you sequence the DNA, only about 1% of the entire amount of DNA is tiger DNA.
Right.
So what we have to do is we have to design experiments in the lab which allow us to only read the tiger DNA. So
Right.
there's an approach called PCR where you basically use two sets of primers.
Mm.
So basically the genome is like a book,
Mm.
and you might want to read word 5,
Mm.
6, and 7
Mm.
in chapter 6.
Okay.
So you basically make little bits of tape which stick to words 4 and 8,
Hmm.
and then basically you're able to read what's in between them. Now,
Highlighting.
yeah,
Hmm.
exactly. So basically you have to find words which are unique to tigers
Hmm.
and not, in all kinds of species. So you
Right.
have to kind of pull out
Hmm.
or fish out the information of relevance, which
Right.
is that of tigers. But you can also use this information, the other DNA, to
Hmm.
look at other stuff. For
Right.
example, we're doing a project on diet,
Hmm.
and we're looking at how tiger diet changes when you go from the core
Hmm.
of a proteidia to the buffer
Hmm.
across seasons and so on. And so we can actually, in that case, we break open the scat, take samples from inside it, and basically sequence the DNA and figure out what tigers are eating in these different locations.
Nice.
You could also try to look for, bacterial load
Hmm.
and whether tigers have particular pathogens or something if you sequence the bacterial DNA, for example, or the
Hmm.
fungal DNA.
Right.
So it's a treasure trove of information,
Hmm.
really.
Right. Could you take us through a typical fieldwork day? How do you go about collecting these samples and ensuring that they're accurate for studies later on?
Yeah, so it really depends a lot on the project, what project we're doing. So, for example, we've been working for many years in Ranthambore in
Hmm.
Rajasthan, where we're reconstructing the pedigree or the family tree.
Okay.
And so there, we don't want to just sample any tiger, we want to sample specific, say, mothers and their cubs.
Hmm.
So there, we have to go into the field, find those individuals,
Right.
and spend the whole day watching them,
Hmm.
identify sites, maybe when they sit down
Hmm.
and take pictures. And once they leave, we go and search those sites for shed hair, for example.
Okay.
In other places, we're studying diet, so we just walk transects, so we just walk in the protected area and pick up samples of fecal
Hmm.
scat or whatever.
Right.
It doesn't really matter whether we see the tigers,
Hmm.
right? We are just sampling along routes, and luckily for us, tigers also use those routes.
Right.
And being territorial, they tend to spray
Hmm.
urine or and defecate to mark that this is my territory. So
Hmm.
that's great for us because then we get the samples
Right.
just by following those routes.
Right. One of the things that really made me curious in your research is your work on the black tiger,
Yeah, yeah, yeah.
which was considered a myth until
Right.
we figured out some kind of mutation in their gene.
Yes,
And
yes,
this again was something that you figured out from studying their poop.
yeah.
Could you tell us more?
Yeah, so basically, it was really, again, I was lucky to be part of this
Right.
story. And, for a long time, people talked about this black tiger. And in fact, it was the native people, the tribals that lived in Simlipal who talked about it. But everyone dismissed their claim, saying, oh, you're just lying. They never
Hmm.
believed them
Hmm.
until in 1993, when in self defense, a teenager actually with a bow and arrow killed a black tiger. He
Okay.
was being attacked.
Hmm.
And that was the first proof that this beautiful and different animal existed,
Hmm.
right? Unfortunately, that skin was burnt down in the fire of the Natural History Museum of Delhi.
Okay.
But, we were approached by some forest department officials from Odisha saying, why does this tiger look like this? Do you
Hmm.
have a clue?
Hmm.
So actually, we were lucky. So it's often very difficult to study a phenotype, appearance,
Hmm.
and its genetics in such an elusive and wild species.
Right.
But what happens in the black tiger is it's a disruption of the striping pattern.
Hmm.
So basically, instead of having regularly laid stripes, they're all array.
Hmm.
And such phenotypes had been studied in cats and in cheetahs.
Hmm.
And so earlier researchers had found a specific gene called TACPEP,
Okay.
which is a transmembrane peptidase. It's a protein, which is mutations in which result in pattern disruption phenotypes,
Right.
like screwed up patterns
Hmm.
for cats and cheetahs.
Hmm.
So we had a candidate gene and we had a phenotype, but we didn't know whether they were linked in tigers specifically. So again, we were lucky that in the Nandankanan Zoo, there was suddenly, just when we were working, there was a litter
Okay.
which included some black tigers,
Okay.
individuals. So we were able to take that pedigree, the
Hmm.
family, 3 generations, sequence their genomes, look at this
Hmm.
gene, find a specific mutation which correlated with this phenotype, and
Right.
then go back to Simlipal, go back
Hmm.
to the field and collect scats of unknown tigers. We
Hmm.
don't know how they look,
Hmm.
identify individuals, and look for that gene, look for that
Right.
mutation.
Right.
Yeah.
Yeah. And I think one of the things you do is also go back
Hmm.
to study how tiger populations have
Right.
declined.
Yeah.
And you identified that 200 years ago there was a significant drop. How do you go back when you don't have live samples?
Right. Right. So basically, that is um, tools of evolutionary modeling,
Hmm.
where you say you have specific amount of genetic variation today, so what processes from the past could result in the amount of genetic variation you have today. So you can model, bottlenecks or changes in size, and you can see what types of the past,
Hmm.
scenarios of the past, correspond best to the observations of today.
Right.
And that's what we had done at that time. It was many years ago and was our first one of our first research projects. But now we are actually, I was just telling you, we are actually sampling trophies from royal families and others who were part of hunting or of tigers. It was a big thing in the past.
Correct.
And so we are able to go back in time literally and get DNA from these small bits of skin or whatever, um, more than 100 years old. So then now
Amazing.
that we have those data, we can actually better, you know, like investigate these models of decline. Was it 200 years ago? Was it 100 years ago? And maybe I think it's going to be much more nuanced, that we will find different phases of decline.
Right.
But we'll see what we find. Yeah.
One of the things also is that despite this decline that
Yeah.
happened, uh, your studies found that Indian tigers have actually a higher genetic variation and could hold the key for tiger survival elsewhere.
Yeah. Yeah.
What is the genetic variation? What did you find
Yeah.
out?
So imagine, like we just said, that the genome is like a book, right? And there's lots of words in the book.
Right.
So if you have, like, a book for every tiger in India, the number of words and, like, the basically the variation, the gene pool variation across India is very high, right? And when you compare it to outside of India, whereas, where tigers could be from the Russian Far East all the way down to Southeast Asia, if you even combine all of that, more than 70% of global genetic diversity is in India.
Okay.
And, today, 75% of global tigers are in India. So, both in terms of numbers and in terms of genetic diversity is like money in the bank in a way, right? It's like potential for the future as well. India holds the reservoir of
Right.
both of these, and so that's why I think India is really key to tiger survival. Also because of the commitment that the government of India has made to tiger conservation, and several recent efforts like the Global Tiger Forum, the International Big Cat Alliance. So India is also, in terms of action, trying to lead conservation, through partnering with countries in Southeast Asia and so on.
Right.
So yeah, so those three axes in terms of the numbers of individuals, the genetic variation, and how we actually save and increase tiger numbers, I think India can lead.
Right. The genetic variation, does it have something to do with how connected tigers are? And if it does, then what about tigers in Ranthambore that you said are probably more isolated?
Yeah. So basically, the fact that we have a high total amount of variation is because probably until recently there were lots of tigers in different habitats which were all relatively connected, right? So the total then becomes much higher. uh, Already we have some, and still some landscapes like the Central Indian landscape, the Western Ghats landscape, tigers are still connected. But there are already some populations which are isolated, like Ranthambore, also Odisha, Simlipal. And so there we see low genetic variation, high inbreeding, or effects of mating between relatives. What we are predicting is there may be inbreeding depression or less fit individuals over time. Now the way to remedy that is to bring in genetic variation, and this is a concept called genetic rescue, where individuals from other landscapes are introduced into those populations. Now that's of course only the genetic aspect. There's
Okay.
many behavioral aspects. Will the animals fit in? What will they do? And so on.
Right.
But that is the way forward for those populations, and that's something we're also working on now.
In terms of understanding the impact of climate change
Yeah.
on biodiversity loss, would your studies of, say, going back in time, how would that help? Would it help us understand how climate has changed over time? What do you kind of seek to understand from that?
So I think with climate, in the context of India, the spaces where climate will directly impact biodiversity are mountains, right?
Okay.
Apart from other places, but a dramatic place. For example, the Himalaya,
Mm.
where, you know, I mean, they are dramatically, so basically, impacts of climate change are much higher in tropical high elevation areas like the eastern Himalaya,
Mm.
right? It's very tropical because it's much closer to the equator.
Right.
And yet, there's ice and you have very high elevations. So long back, I've done some work on species like pikas, which are high elevation specialists, and predicting that as temperatures warm, what might happen to the distributions of these species. Will they be pushed?
Mm.
Where will they go? And for species that live at high elevation, there's nowhere to go,
Mm.
right? And this is also true similarly for montane or high elevation species in the Western Ghats.
Mm.
So they will directly face impacts of climate. Overall, the predicted effects of global change in India are several, as you may know, but they include changes in predictability of rainfall, extreme events, warming, of course, right? And to mitigate these detrimental effects of climate, the best solution is nature,
Mm.
right? Protection of biodiversity. While we may say, so why do we need to conserve biodiversity in central India? Is it
Mm.
just for tourism so that
Mm.
people can see tigers and other wildlife? But intact ecosystems anywhere are mitigate against the effects of climate, okay,
Mm.
and provide services like pollination and so on, right?
Correct.
So I think that going forward, the big challenge is going to be, how do we mitigate climate changes upon us?
Mm.
And forget about biodiversity, humans are being very, very badly impacted, right?
Yeah.
So how do we mitigate these effects going forward? Strategies like restoration, where we are trying to recreate through action like
Mm.
management, relatively intact vegetation and biodiversity, rewilding, bringing back species to areas where they were not, and also thinking very uh, deeply about what the ecosystems were. So, for example, a large part of India is arid,
Mm.
and yet the continual tree planting kind of tips those ecosystems more towards forest like versus savanna like
Mm.
ecosystems. So, several species, for example, tigers are now, I mean, of course, they have much a way to go, but species that are dry zone
Mhm.
species like wolves,
Mm.
are doing really badly. So, I think that uh, it's a package. Conservation and climate uh, is a package.
Mm.
It's not different.
Correct.
Climate mitigation, detrimental effects of climate change will only be mitigated by nature. I hope that makes sense.
Yes.
Yeah.
And does how much does your genetic study reveal about the actual impact of climate change on tigers or other mammals like wolves that you
Right.
mentioned? Yeah.
So we haven't explicitly explored that for these large
Mm.
carnivores. We're just starting some work. When we did some work on a global tiger range, we did find, for example, uh, cold associated adaptations in
Mm.
Siberian tigers.
Okay.
So basically, what would happen is, over time, species which live in certain environments will have adaptations for those environments. Now, when the environment changes, they either habitat track. They
Mm.
try to go to where the environment is ideal for them,
Mm.
or if they just stay where they are. Now, the problem is, if those adaptations make it worse to live
Mm.
where they are.
Mm.
And for many species like pikas or yaks, or these high elevation specialists,
Mm.
that is the case.
Mm.
But for these generalists, like
Mm.
say tigers that live in many habitats,
Mm.
we are not sure.
Okay.
And I think we need to do a lot more research to
Mm.
understand those things better. But definitely, these historical materials have been used, for example, in the US,
Mm.
to understand the impacts of climate on species. So it's possible,
Interesting. Mm.
but we'll need more work to understand that better.
That brings me to actually the policy aspect of it.
Mhm.
A lot of scientific work tends to remain in the theoretical realm when it comes to
Right.
application, but your work actually translated into redesigning an entire national highway because you found how tigers were traveling between two national parks. Could you
Right.
tell us more about what you found and how that impacted the change in policy?
Yeah, yeah. So basically, I think maybe we should dis- distinguish between action,
Hmm.
management, and policy.
Right.
So policy is a very big thing,
Correct.
right? I think that uh, the lot most of the scientific work can actually impact action
Hmm.
or management. Say do this versus that, something like that.
Right.
And in the case of this highway, we showed there was connectivity between Kanha and Pench. These are two protected areas in MP, Madhya Pradesh, and Maharashtra. And at that time, there was a plan to widen a National Highway 7 from a 2-lane to a 6-lane.
Hmm.
And basically, we actually did simulations in our paper which showed that, should this happen and this highway become a barrier, the probability of extinction,
Hmm.
because this whole landscape of tigers is maintained by them moving from one location to the other,
Right.
and the process of extinction will be accelerated
Hmm.
if this becomes a barrier. So this and other work, not
Hmm.
just our work,
Hmm.
actually was taken up by a citizen in
Okay.
a public interest litigation
Okay.
against the NHAI, National Highway Authority of
Hmm.
India. So it's interesting. Like I just did the work. We just
Right.
did the work. I mean, me and my students.
Hmm.
I don't do anything by myself. And we published the paper, and someone else took it up,
Hmm.
right? And basically argued in court. It went to the Supreme Court, and basically NHAI was mandated to build this underpass.
Hmm.
Since then, of course, there's been a lot of work by the Wildlife Institute of India and others showing that this underpass actually works and animals are moving. So
Right.
that is an example of a conservation success. And since then, also there was documents presented at the National Board for Wildlife that so basically a lot of wildlife policy or laws are for what's in protected areas
Hmm.
or what are protected species, but the outside is kind of up for grabs,
Hmm.
right? So uh, a lot of discussion about how do we plan corridors, how do we look at the outside parts of protected areas and so on, landscape planning. So affecting policy takes a long time,
Right.
but if we can actually do work which informs on ground action over time, there is a build up of a body of knowledge which
Hmm.
impacts policy.
Right, which is my next question. Is that do you think public-private partnerships or are ways for academic work and research to actually reach a stage where people are discussing it at a policy level?
Yeah, so definitely I think that I have found that what helped me uh, is communication. So
Hmm.
basically, we work in the outside world.
Hmm.
So for a lot of people, you work in the lab, you publish your papers, you don't get an opportunity or necessarily a means to communicate with the outside world.
Right.
But my work is outside,
Hmm.
right? We work outside,
Correct.
and so we have to communicate with people to just do our work,
Correct.
and that builds this how should I say, this habit of communication, in a way,
Hmm.
of communication with different kinds of people, not just
Correct.
scientists, right?
Correct.
With bureaucrats, with people from local communities, with all kinds of people,
Right.
right? And that communication is good because it challenges you. You have a set way of thinking,
Hmm.
and then someone asks you a question and you rethink,
Hmm.
just like teaching.
Right.
So I think that communication, effective communication, can I don't know, I won't say effective, actually just communication
Right.
um, is a really good tool uh, to basically get the word out, and that makes a huge difference.
Hmm.
I have found getting the word out makes a huge difference.
Right.
So I think that that is where there can also be partnerships.
Hmm.
The other place where there can be partnerships is in terms of technology.
Hmm.
So technology is changing very rapidly. Many times, as a scientist, we are always looking for technological advance from the West. But if there are technological advances here, which we could, leverage
Right.
and work together, that would be a really nice opportunity for partnership as well. I
What
hope that
would,
answers your question.
yes,
Yeah.
what would some of these technological, what is a wish list, a technological
Oh,
wish list that you would have for your
yeah,
work
yeah.
specifically?
So basically, like a lot of the work we do, we bring the samples, we come back to the lab, and then we work on them here.
Yeah.
There's some really cool companies which are making like field ready labs,
Nice.
like
Okay.
bento labs,
Mm-hmm.
and other companies And they've also made sequencers, which are handheld sequencers. Of course, that's a very big thing, nanopore, for example. But small things like we could customize, like mobile labs, and actually work with them in the field, but also use them as ways to communicate. There used to be this really cool initiative called the Science Express, which was by the Department of Science and Technology, where they had this train outfitted with all kinds of outreach material on science, and it went across the country and it stopped in every single a station, and then the kids from all over would come and see all this stuff.
Right.
So ways to actually have mobile, because I mean, cities are one thing, people have a lot of exposure, but in a lot of other places exposure is not as much.
Right.
And who knows, there may be so many amazing young scientists out there who just didn't know that they could pursue this career path. So it'd be really fun to actually work to democratize science and technology. Would be really cool.
Right. And finally, the last question is there a research question that kind of keeps you up at night, that you think needs more work?
Yeah, so I guess the thing which really keeps me up at night is trying to understand. I mean, it's kind of maybe a little technical, but you know, uh, we've always said that genetic variation is really important for survival, and yet we see so much resilience in our animals. Uh, you know, uh, I mean, animals are amazing, right? So how are they, like, I don't know whether we'll ever understand that, because we only have ways, like we can only interrogate things with the tools we have.
Right.
How do we understand how animals think? It's like very difficult, no?
Yeah.
So I would love. I mean, I think that's what keeps me up these days, is thinking about how are animals so resilient. They know when to come out, when not to come out, where to go, what to do. They hide so well.
Right.
They survive so well. Look at elephants. I mean, they're like amazing, right?
Yeah.
So animal cognition, how do animals do what they do? How are they so resilient? uh, and I just worry sometimes about what the future will look like. But I guess, given animals are so resilient and people in India definitely very accommodating, hopefully it'll be a
We'll
good
find a
future.
way. Thank you so much for your time. It was amazing meeting you and for your space here. Thank you.
You're welcome. Thank you so much for coming and having me.
Thanks. If you like this episode, subscribe to us on YouTube and wherever else you get your podcast.